So while it may be easy to understand how selectively neutral stretches of DNA can persist in a population, we're still stuck with the problem of how is it that recessive alleles are able to persist if they're subject to negative selection. So first let's look at what happens if indeed our recessive alleles are somewhat damaging. Once they become rare within the population, they can hide in a diploid. The allele will actually be hidden from selection because it's only found in the form of a heterozygote. So let me give you an example. Here's a really, really nasty, lethal mutation. So this is called human autosomal dominant polycystic kidney disease, so abbreviation ADPKD. It causes kidneys to get large. They have these horrible cysts, okay? And if you have one copy of the capital A allele, if you're even just a heterozygote for that, you die because the kidneys are just so deformed that they can't function properly, okay? Now, if there's an allele that is that lethal and it's dominant, it simply cannot persist in the population. Everybody who possesses it dies in infancy or even before birth, so that you will not find this in the adult population. So this is a case where the allele is lethal and it's dominant and it disappears entirely, okay? You only see this dominant lethal allele by new mutations. And so this particular genetic disorder is found only to afflict about one child out of every 100,000 births. So that reflects the underlying mutation rate at that particular stretch of the DNA. Once it's in, it's gone. So it can never persist. It's awful. But now let's contrast that with a lethal recessive. Here's another inherited disorder, a genetic disorder called cystic fibrosis. Cystic fibrosis, I mentioned a lecture or two ago when I was talking about frame shift mutations. This, again, causes terrible health problems, a thick mucus in the lungs, and it really compromises the immune system. And it's terrible, but it's only terrible if you happen to be homozygous. It's a recessive allele. If you have two copies of this defective allele, then it's essentially a death sentence. But the heterozygote is fine. Heterozygotes do not suffer any reduction in their survival or reproduction, okay? So we have this allele that is absolutely deadly, but it's recessive, so only the homozygote recessives suffer from it, okay? So we have then in a population, anybody who just has one copy of it, the recessive allele is hidden. So a man or a woman can be a carrier for cystic fibrosis, and unless they marry somebody else who's also a carrier, the allele will continue to be hidden. But there are certain populations where the allele is somewhat more common. And so, whereas about 1 in 4,000 American children are carriers, that is, they're these heterozygotes, and it's very unlikely that any two American children at random who are carriers will meet up and have children. It's pretty unlikely that you get cystic fibrosis in your family. But if you're descended from Jews that originated in Europe, that population has a much higher gene frequency for this particular allele. So 1 in 20 European Jews are carriers. So it's quite common in Jewish communities that couples, when they get married, there's a pretty good chance that 1 in 400, that any couple, both of them will be a carrier for the allele. And if they're both carriers, then yes, a proportion of their descendants will be safe because they'll be homozygous for the healthy allele. Half will be heterozygous, just like the parents, so they'll also be carriers. But there's a 1 in 4 chance that one of their children will have cystic fibrosis, okay? And so there will be tests often encouraged in this community to look for signs of the CFTR allele, which is coded on chromosome number 7, to see if there's a risk that they might have children that would be born recessive, homozygous recessive for cystic fibrosis. So in the case of these recessive lethal alleles, as we've seen, lethal recessive alleles are only visible to selection when common enough for two heterozygotes to meet and produce homozygote offspring. So amongst American families, only when both parents are Jewish is there a serious risk of producing a homozygote offspring. For everyone else, the risk is very, very low. And so really, for the whole population, these alleles may be present across hundreds of millions of people, but they're almost always hidden because they're only found in heterozygotes. And the heterozygote doesn't suffer any reduction in fitness compared to the perfectly normal individual. So lethal recessives, once they get rare enough that they're only found in the heterozygote form, they persist at very low frequencies, and the only time we really expect to see these come back in, besides if you have a founder group like the Jewish American population, would be through close inbreeding. Anyone in your family may carry one of these rare alleles by common ancestry. Because you both have the same father, there's a chance that both of you will carry the same allele through common descent. And so this is again why inbreeding is not a good thing. And just to give you a rather sublime example of what happens with extreme inbreeding, let's go back to the royal family in Europe yet again. Now we're going to look at the Habsburgs. And what we want to look at is the ancestry of Charles II, who was king of Spain, I believe. And Charles II lived from the 1600s to the year 1700, okay? And if you go back in his ancestry, he's got ancestors who were from Austria. They were back in Holy Roman Empire, Portugal. So all of these family trees are well documented, okay? And if you go back the preceding 200 years, 9 of 11 marriages in his family tree were between cousins or between uncles and nieces, okay? That's a lot of inbreeding. So there's a real chance that only one of these individuals may have been a carrier for some nasty allele, but then multiple descendants would all have it. They then marry each other, keeping it around, and it might build up more and more of these homozygote recessives. So by the time we get to Charles II, generation after generation of that kind of inbreeding, he was a sorry sight to behold. Charles II's tongue was so big for his mouth that he had difficulty speaking and drooled. He also suffered from an oversized head, intestinal upsets, convulsions, and, according to his first wife, premature ejaculation. He was unable to speak until the age of 4. He couldn't walk until he was 8. He was short and weak, with little interest in his surroundings. He looked old when he was 30, with swellings on his feet, legs, abdomen, and face. He could barely stand and suffered hallucinations. So again, close inbreeding is a very bad idea, and the reason is the greater chance that the descendants will end up being homozygous for these rare recessive alleles.